Let's look at a second line of evidence for evolution having occurred. And again, remember, by evidence for evolution, we're looking for evidence that suggests that existing species today have come from pre-existing species in the past and have diverged over time in terms of their DNA and their phenotypes, etc. So we're looking for common ancestry, evidence of common ancestry to existing species. So here we're going to look at what might be referred to as molecular phylogenetics. Phylogenetics. So we've talked about phylogenetics in another tutorial, and that's looking at the history of various species over time from common ancestors based on things like morphology, that's the form or makeup of the body as a whole, or their structures. But it can also be based on biochemistry, that's the way the body functions in terms of chemical reactions, or it can be based on genetic sequences. So molecular phylogenetics looks at evolutionary relationships based on molecular evidence. And there are several strands today of molecular evidence which have proved very powerful in looking at relationships between species through history. And one of the common ones, and one of the first ones to be used, was DNA hybridization. And I'm sure you'll look at this process and it will make a lot of sense to you, since we've talked about it in relation to genetic engineering and other kind of applications. But here it's being used very powerfully to look at the number of bases in DNA sequences that are held in common between different organisms. So hybridization means to join DNA from two different sources, and in this case it's from two different organisms. So it's outlined here below. So if we look at, here's organism A's DNA, double-stranded DNA, here's organisms B DNA. So they're denatured, that is, they're heated to separate the strands. You'll be familiar with that process from the polymerase chain reaction. So we end up with the DNA separated into single strands, and then they'll be mixed together. So you combine the single strands from one organism, A, with the organism B. So mix the strands together. Then, of course, you'll cool the DNA, and where complementary bases can line up, they will naturally form hydrogen bonds and rejoin. So, of course, if you had two organisms that come from exactly the same species, that is, they have the same genomes, you would expect a 100% or complete hybridization because they're the same species. If you get partial hybridization, so some parts of the DNA join back together and other parts don't, then it would suggest that the organisms are related, because it would make sense that if they had evolved from a common ancestor, that they would actually carry over some of the DNA from their past into what they are today. So if the organisms are related, you'd expect them to have certain sequences in common. And this is one of the ways you can work out the percentage similarity between species and how far apart or how long ago they diverged from a common ancestor. Of course, if there's very little or no hybridization, you would have to say that the two species are completely unrelated, and by completely unrelated we mean going a long way back in time since their common ancestor, and it may be, tens of millions of years you're talking about here. Keeping in mind that nearly every organism is related in some way to another, but if you go back in an incredible distance in time, there may be very little, if any, of the DNA sequences left in common. But the more recently organisms have diverged from common ancestors, then the more DNA you'd expect them to show in common. So this has become a very powerful method of working out evolutionary relationships. If we look at this diagram here, and there'll be similar ones in your textbook and Biozone manual and other books, you can, evolutionary biologists can estimate or work out roughly the rate of DNA change, that is the rate of mutations that can occur in something like mitochondrial DNA or even nuclear DNA over time. So one mutation every so many divisions or replications of the DNA over time. They can work that out, with reasonable precision. So they can translate something like the percentage difference in the DNA into millions of years since the divergence from a common ancestor. Now remember, we've looked at cladograms in another tutorial. If you look at this cladogram here, each of these, of course, is a speciation event where new species diverge from a common ancestor. And the question, of course, here is, when did these speciation events occur? Well, just keep in mind, you can never say precisely. You're dealing with millions of years and you're dealing with data that can be a bit fuzzy at times, but nevertheless, what's fascinating is that there is a reasonable degree of accuracy in what they can say here. So if you look at the primate separation, you can see, for example, the common gibbon and the Siamang gibbon, which we'll probably see in the zoo in a week or so's time, diverged here more recently, okay, than the gibbons did from the old world monkeys. Okay, so this speciation event here happened about 20 million years ago. The common Siamang separated from the the common gibbon separated from the Siamang gibbon about 7 to 8 million years ago there. And then along a different line of evolution, you can see where the things like the orangutan, gorilla, the human and the pygmy and common chimp diverged from the line of evolution. So this is how DNA hybridization can be used as evidence for molecular phylogenies and working out the relationships between different species.